Hey everyone, this is Crystal Abbey, your instructor for Biology 1111. Um, Today we're going to be covering Chapter 6, which is Energy for Life, which is primarily about photosynthesis, which is the type of energy process that occurs in organisms that have chloroplasts. Things like plants primarily is what we'll think about when we think of chloroplasts. So let's look at an overview of what photosynthesis is. So what it does is it transforms solar energy, which means it comes from the sun, into chemical energy in the form of carbohydrates. Okay, so photosynthesis harvests energy from the sun to produce carbohydrates. Um, the next thing you'll see here is our chemical equation for photosynthesis. You probably would do well to remember this equation here because you'll wind up being able to answer a lot of questions about photosynthesis from knowing this equation. It'll also help you for next chapter too when we talk about cellular respiration. But our chemical equation for photosynthesis is 6 molecules of carbon dioxide plus 6 water molecules plus solar energy yields glucose and oxygen, 6 of them to be specific. So the organisms that do this process of photosynthesis are, of course, our plants, but also other organisms such as algae, as well as cyanobacteria. So they're all special and they have chloroplasts or chloroplast-like structures in bacteria that enable them to perform photosynthesis, which is transferring solar energy essentially into a chemical form, which would be the carbohydrate, glucose. All right, so these organisms we refer to as producers. We call them producers because they are able to produce their own food. They feed themselves. You don't have to feed a plant. They are capable of harvesting that energy from the sun to make its own glucose, to make its own food. Additionally, we call them producers because they help produce food for consumers. So consumers are organisms that consume things. For example, you and me, we're consumers. We consume stuff in order to harvest the chemical energy from it. So, for example, if I were to eat a handful of almonds, those almonds are they derive from a plant that is a producer that produced its own food and also produced food for me as a consumer. Um, they also are going to produce the food for pretty much the majority of other living things on Earth. Any food chain always goes back to some kind of producer. So, for example, if we have, a human, that's, me and you, we ate a deer. I'm not going to try to draw a deer because it will be awful. But eats, that deer eats plants. It's an herbivore. And that plant got its energy from the sun. And no matter what food web, there might be very few exceptions, but pretty much the vast majority of everything is going to wind up ending with sunlight. Okay, so we'll talk about plants for the most part in this chapter because they are the most, commonly associated organisms with photosynthesis. Okay, so the areas of plants that are involved with photosynthesis are primarily the green portions of plants, so particularly the leaves of plants. So this image over here to the right shows you just a typical leaf structure, and if you take this small section and look at what it looks like on the inside, this is what it shows you. So we have this mesophyll tissue here, which primarily houses the areas of the plants that perform photosynthesis. Leaves have veins, similar to how our bodies have veins and arteries that help transport stuff throughout our body. Plants have similar structures. They're called phloem and xylem that allow for nutrients and water to pass through to direct it to various portions of the plant. Okay, another structure you need to be familiar with here is the stoma. All right, so bear in mind that stoma is singular and stomata is plural. So you'll see they wrote stoma here, and it's pointing to that little structure. And I've written stomata here. Again, same thing, they're just one's singular and one's plural. Stoma is singular, stomata plural. But these are little tiny holes that are located on the undersides of the leaves that act kind of like quote unquote plant mouths. So just like our mouths allow us to breathe in oxygen and breathe out carbon dioxide, they do the reverse. They breathe in carbon dioxide, as shown here. You can see CO2 going in and breathing out oxygen. Some other structures of importance with photosynthesis are the roots of plants. Now, the roots are not shown in this image, but we probably all know that the root system of plants are found underground, and their function is to absorb primarily water, but also nutrients from the soil. Now, carbon dioxide and water that can diffuse into these mesophyll tissue cells and then into chloroplast, which is where the actual photosynthetic process takes place. So again, carbon dioxide will come in through the stomata, water will come up through the roots of the plant all the way to the leaves and enter into these mesophyll cells. And in those mesophyll cells are where our chloroplasts are located, where they're going to be needed. Here's the general structure of our chloroplast right here. So we've talked about the chloroplast before and how they have this double membrane layer. You can see the one on the outside and then there's one on the inside here. And this inner layer helps to form these disc-like structures in here that look like stacks of coins. If you'll recall from a previous chapter, we said that these discs are called thylakoids, and when they're stacked like this, we call the stacks grana. And this shows a transmission electron microscopy image right here. All right, so this just goes through chloroplast structure. Again, it's a little larger picture, but of course, again, it has our double membrane. The internal membrane has these membranous sacs or discs that we call the thylakoids. The thylakoids are hollow, right? And don't forget, they're also made up of a lipid bilayer. Remember, just like they're a cell membrane. And it has a space in here that we call the lumen. The stacks of thylakoids we refer to as grana, excuse me. And another term of importance is going to be the stroma. So stroma is the liquid that is inside the chloroplast. Remember, we said that was synonymous with the cytoplasm of the whole cell. It's just the fluid within. All right, now within these thylakoids, these little disc shapes, within the membranes of them, the thylakoid membranes, they contain these photosystems, and these photosystems contain chlorophyll. Chlorophyll is a pigment. Now, other pigments also reside within thylakoids, and regardless of which pigment we're referring to, they absorb solar energy. And we'll see where all that fits into the big picture in just a minute. Carbon dioxide during the process of photosynthesis will be reduced in the stroma, that's in the liquid, into carbohydrates, glucose. And that glucose molecule is our chief organic energy source for most organisms. Remember, plants also have mitochondria, so they'll be able to use their mitochondria to form ATP from that glucose, just like we do as humans. And we'll talk about that process in cellular respiration in the next chapter. Okay, so the photosynthetic process begins with the end products of cellular respiration, and that whole idea will really come to when we get to the next chapter. What you'll see is it's kind of a cycle where the end products of cellular respiration, which is what you and I do because we have mitochondria, are the beginning product or the beginning reactants of photosynthesis and vice versa. All right, so hydrogen atoms. During the process of photosynthesis, the hydrogen atoms are removed from water and are added to carbon dioxide. And solar energy is required to do that. This demonstrates that here. Here we have water and CO2, which is carbon dioxide. And the oxygen that is in, the hydrogen atoms that are in the water molecule are removed and added to CO2. If we take these two hydrogen atoms and tack them onto CO2, well, this is what we get, CH2O. Likewise, we're left with oxygen then. All right, so if you count up all of the hydrogens and the carbons and the oxygens on side one of this equation, it will be equivalent to what's on this side. They're just rearranged. And that's what photosynthesis does. It rearranges these chemicals into something that's utilizable by the cell. All right, oxygen is a byproduct of the oxidation of water, and the end product is CH2O, or if we have enough of this reaction going on, glucose. If you were to take this and multiply the different components here by 6, you would get that. For example, if we take a single carbon, multiply it by 6 more, we get 6 carbons. If we have 2 hydrogens, multiply it by 6, we get 12. And oxygen, multiply it by 6, we get 6 oxygens left over. All right, so this process occurs in 2 phases. They're called the light-dependent reactions and the Calvin cycle. The light-dependent reactions happen during the daytime because they depend on the ability of the availability of light. So, of course, during the nighttime, the light reactions won't happen. They occur in the thylakoid membrane. And in this phase, light is absorbed by chlorophyll, which is in the photosystems of the thylakoid membrane, and that light is converted into chemical energy. And then oxygen is released through the hydrolysis of water. So this image actually shows you that here, and we'll talk about all these little things as we move forward. But again, the light-dependent reaction, here's phase 1. Calvin cycle is phase 2. The light-dependent reaction depends on light. Water is an input, and oxygen is an output. We have 2 more outputs, NADPH and ATP. We'll talk about those later. I don't want to get to those just yet. I want you to see the big picture first before we get into these little details. Secondly, the Calvin cycle is going to utilize the ATP and the NADPH to fuel its furnace, as well as an input of CO2. And again, CO2 came in through the little plant mouths, stomata. Water, it comes from when it rains or if you water your plants. As an output of the Calvin cycle, we have our sugar, which is glucose. Or we can also say our carbohydrate. Remember, carbohydrates are just chains of sugars. And then as an output, we also have NADP plus and ADP, adenosine diphosphate with an additional phosphate group. All right. And again, this is a cycle, right? So, and this whole thing is kind of a cycle too, because, you think all these things that are going on in the light reaction, some of the things that are produced are fueling the Calvin cycle, where they are then broken down to ADP and NADPH, NADP plus, which is then reused to fuel the light reaction again, and so on and so forth. So again, 2 phases, light-dependent reactions and the Calvin cycle. This picture shows the same thing. Again, we're in our plant cell here. We're looking inside of a chloroplast, which you can see bunches of them over here, the green stuff. And then this shows you one that's really blown up very largely. So we input solar energy. We're inputting water from the roots. And in the light reaction process, we're expelling oxygen, which will then be breathed out by the plant through the stomata. Then as a byproduct, this process will also yield ATP and NADPH that will then go on to fuel the Calvin cycle reactions. In addition to that, we'll have CO2 that will be a player here, and together they will produce glucose, or the carbohydrate, or sugar, whichever you want to say. And as a byproduct, they will break down NADPH and ATP into ADP and phosphate, as well as NADP plus, that can then be reused to do another round of the light reactions, and back and forth it goes. Okay, so Let's go through this a lot slower and a lot more in detail now, because I know it might seem a little, this whole process might seem a little overwhelming to you. Make sure you go through this PowerPoint ideally a few times. Watch the videos that I post on it, because they're going to be really helpful for understanding it. You'll probably have to spend a little time on it by yourself, reading through the step-by-step processes in order to really truly follow it and understand it. It is a complex process. All right, so at the very beginning, how this happens is our pigments absorb solar energy. Pigments. Now, pigments are molecules that essentially can be seen as color. So solar energy can be described in terms of its wavelength and energy content, and you're going to see why that relates to pigment in just a minute. So solar energy can be described as visible light, and it's a small section of different wavelengths that fall on a larger scale of the electromagnetic spectrum. Shorter wavelengths contain more energy, and longer wavelengths contain less energy. And only about 42% of the solar radiation that reaches the Earth hits the surface. But vision and photosynthesis, both of these two, that's why we call it visible light, are adapted to use the most prevalent wavelengths that hit the Earth. And so let's take a look at what we actually mean by light. We said it has to do with wavelength, but what is a wave? We'll get to that in a second. But light is energy. It's not just used to see, but it is in a form of energy. And like any other energy that we've discussed, it can travel, it can change form, and it can be harnessed to do work. Just like when we talked about potential energy and kinetic energy, and we've mentioned chemical energy, which is a type of potential energy, they can all go from one type to the next. Same thing is applicable with light energy. And light is a wave that falls on the electromagnetic spectrum. Now, when we talk about wavelengths, what we're referring to is the distance between two consecutive points on a wave. So waves are going to oscillate up and down just like this one, right? And if we measure the distance from point A to point B, that's what the wavelength is. We can measure wavelength from the very top of this wave to the very top of the next one. That's called measuring from crest to crest. Think of it like the crest of an ocean wave. It's at the very top. We could also measure wavelength and get the same result by measuring from trough to trough. Trough is the very bottom. If we measure the wavelength from crest to crest, it's going to be the same as trough to trough. Also, I mean, it really doesn't matter where we start. As long as we end at the next consecutive point, we'll get the same answer. So if we measured the wave here from this distance to this distance, then it would be the same as measuring it from trough to trough or from crest to crest. But that is the wavelength. It is a length of measurement. Some waves are really, really long, and some waves are really, really short. Now, long wavelengths, of course, then will have longer distances between two consecutive points. So here we see if you were to measure from this point to that point, here to here, it would be relatively much longer than measuring from this point to this point, correct? So this wave has a longer wavelength than this wave. Okay, so like I said, these electromagnetic waves are going to fit in the electromagnetic spectrum. The electromagnetic spectrum contains all of the wavelengths that are known. Visible light is just a small portion of it. So that is shown here. On this end of the spectrum, we have really, really long waves. And over here, we have really, really short waves. These radio waves can be like kilometers long. That's extremely long. And these gamma rays down here are super, super tiny waves. Very, very dangerous, actually. Much more dangerous than X-rays, for example. Y'all are probably familiar with what X-rays are. So here we have UV radiation, which is a wave as well that can be dangerous and harmful to humans. On this side over here, we have infrared, micro, and radio waves. So again, wavelength increases on this end of the spectrum and it decreases on that end of the spectrum. Now, somewhere here is where our wavelengths are that coincide with visible light. And this little area of our electromagnetic spectrum is blown up here so you can see it really well. But waves that are that have wavelengths of 380 nanometers up to about 750 nanometers. Still really, really small. Okay, so each one of these wavelengths also are going to correspond to a different color. So for example, over here at 380 nanometer wavelengths, we see a purplish color. And then as we move to larger wavelengths, we begin to see blue, green, yellow, orange, and red. So our photoautotrophs, the plants, the organisms that make their energy from the sun, capture light energy from the sun that falls between the this visible light spectrum. And again, it's due to pigments. So how do these organisms absorb light? And again, it's due to these pigment molecules. One of the most commonly known pigment molecules is chlorophyll. There's different types of chlorophyll. We'll talk about two of them, chlorophyll A and B. Chlorophyll A and B absorb light, which are again are waves, that are going to coincide with violet colors, blue colors, and red colors. I want you to look at this table or the graph here on the right. So this is showing you absorbance of wavelengths, and this shows you the wavelengths at the bottom. So here we have, our violets, which is around 380, all the way up to our reds, which is around, 750. So you can see absorbance of light waves are very high in this area, from around 380 to 500 and around 500-ish. And then we see a large dip, right? So in this area here, there's not a whole lot of absorbance of wavelengths. And if you'll notice, those wavelengths coincide with a greenish, somewhat yellowish color. So if an organism cannot absorb that wavelength, then it's going to reflect it. And then that wavelength that is being reflected is going to come into our eyeballs and then be transferred, that signal will be transferred to our brain, and that's why we see plants as green. So again, either you absorb or reflect wavelengths. If it's absorbed, then we won't see that color. We only see what is reflected. So again, because in this area we see relatively low absorbance, then it must be reflected instead. And because it is associated with green-ish, a little bit of yellow over here, but primarily green associated wavelengths, that's why we see plants and their leaves as green. So you're probably thinking, okay, well, what about plants that aren't green? What about fall colors, for example? The pretty leaves starting to turn yellow and orange and brown. And the reason why that this is possible is because chlorophyll A and B are not the only pigments. There's lots of pigments that are found in plants. Chlorophyll A and B are just two of the very, very prominent ones. Carotenoids are also found in pretty much all plants. They're another type of pigment, and they have absorbance patterns that are different from chlorophyll A and B. So carotenoids, they absorb light in violet, blue, green range, and they reflect the yellow and orange wavelengths. And so that's why we see them as orange and brown and red. Right? So you can see that absorption pattern difference between chlorophyll A and chlorophyll B, and the carotenoids, the line for carotenoids is in orange, is very different. They absorb all this green, blue, and violet and reflect yellow, orange, and red. Okay, so how does this light-dependent reaction work? Now that we understand exactly what light is, we will talk more about the light-dependent reaction. So the light-dependent reaction kind of works like solar energy panels. So solar energy panels harvest energy from the sun and they store it in the form of a battery. Of course, plants don't have batteries, but it's kind of like that. They harvest solar energy and they store it in the form of a hydrogen ion gradient. And so if you'll recall from last chapter, a gradient is where we have, this is my phospholipid bilayer, but a gradient is where we have a difference in concentrations on one side of the plasma membrane than the next, or the other side. So here you'll see there's lots of hydrogen ions, very few hydrogen ions on this side. That is a concentration gradient. Now that gradient is used to produce ATP and NADPH. And the way it does that is by utilizing photosystems. More specifically, it's two photosystems. They're named photosystem 1 and photosystem 2. And these photosystems contain pigment, like chlorophyll, like carotenoids, and they also contain an electron acceptor. So this complex, this photosystem 1 and 2, they serve as kind of like an antenna for solar energy. So we have these solar energy waves that are coming down from the sun, and they're beating down on these plant leaves, and that photosystem that's within the thylakoid membranes, within the chloroplast of our plant cells, is kind of acting like an antenna, harvesting all of that solar energy and helping to pass that energy into a reaction center. Now, the next thing I want y'all to know is that they are named for in the order in which they were discovered and not the order in which they participate in photosynthesis, because photosystem 2 is actually going to be our first step. It's the first part of the process. So, but I did want to show you this picture before we go any further that elaborates not to forget that this is going on in our thylakoid membrane, which is made up of phospholipids. As you can see, all those yellow tails are the fatty acid tails of our phospholipids, and the little red balls are the glycerol molecules with the modified phosphate group at the end. We talked about their structure a few chapters back. So we can see the bilayer here. It is embedded with, excuse me, our photosystems. Photosystem 2 is first because they are named for in the order in which they were found. And then we have photosystem 1 here. Excuse me. But this is going to, this way that the photosystems are structured, basically next to one another, along with this electron transport chain. You can see that labeled here. We'll talk about what that means in a minute. But the fact that they're all really close together helps to promote really efficient transfer of electrons, as well as hydrogen ions. But also in this complex is this last enzyme at the end, ATP synthase. And you can probably guess what he does if you break it down. ATP synthase will synthesize, it's an enzyme that synthesizes ATP. All right, and we'll talk about the order of flow of all of these things as we progress. But I just wanted to give you a visual representation of what it looks like. We have our photosystems embedded into the thylakoid membranes, PS2, we have PS1, we have an electron transport chain in between, and we have ATP synthase at the end. All right, don't get too overwhelmed with all the different things that are written here. We'll talk about them. Okay, so like we said, the first participant here is photosystem 2. In photosystem 2, we're going to have solar energy input, light's coming in, and it's going to energize electrons, and that's going to make them break free from the photosystem and go pair up with an electron acceptor molecule. We don't need to know the details on their names. It's just something that's going to accept the electron. Then those electrons will be sent through an electron transport chain. We'll get to that in just a second. But before we do, we need to know what's going to replace those electrons that were lost. The way we replace electrons that were lost is by splitting water. This is called photolysis. So in photolysis, water, it's shown here, will split into 2 hydrogen ions and an oxygen. 1/2 of O2 is an oxygen. That oxygen here is a waste product. The plant breathes it out. And then those 2 hydrogen ions will be utilized in some of the next steps that we're going to discuss. Okay, next step we'll discuss is the electron transport chain. So we just discussed what happens here, the previous slide. Now, the next step is the electron transport chain. I'm going to say ETC, electron transport chain, ETC, for short, just to save me from getting so tongue twisted. But this is going to occur in the thylakoid space, as well as in the membrane. So the thylakoid space is a reservoir for hydrogen ions. Every time we split water in that previous step in photosystem 2, we said that there's hydrogen ions that will be released. Well, they're going to hang out here in this thylakoid space. This is all this area here is the thylakoid space. You can see it's written there. This is going to be the stroma. That's on the other side of the membrane. So energy from electrons that are transferred between carriers, those are here in the electron transport chain in blue. Those electrons that were released here in photosystem 1 are now going to be transferred to the electron transport chain and passed down. You can see the flow. It's going that way. And when they do, that's going to allow for hydrogen ions to come from the stroma to this reservoir on the other side in the thylakoid space. So not only are we now having excess hydrogen from photosystem 1's process, but now we're getting even more from the electron transport chain. So we're having a huge buildup of hydrogen ions over here. That establishes a very stout gradient. Again, gradient means there is a difference in the amount or concentration of a chemical on one side of the membrane versus the other. And that is going to result in having a large amount of potential energy. Recall from our previous sections or previous chapters when we discussed this before, where if we have this concentration gradient, it's going to have a large amount of potential energy because our cells want equilibrium. And so in this case, we have lots of hydrogen ions on only one side, and we're not at equilibrium. Our cells want to be. So there's a lot of potential energy here. And so because of that, hydrogen ions, they want to get to the other side. They want to go back to the stroma to where we can equilibrate how many hydrogen ions are on either side. And so to do that, these hydrogen ions will be passed down ATP synthase. And every time a hydrogen ion is passed down ATP synthase, we rejuvenate ADP plus a phosphate group back into ATP. So remember, adenosine diphosphate, there's 2 phosphate groups. If we add that extra phosphate back on, we've regenerated ATP, adenosine triphosphate. Now, this ATP here, you'll see on the next upcoming few slides, when we talk about the Calvin cycle, that ATP will be used to fuel the Calvin cycle. All right, but before we get there, we can't forget photosystem 1. So photosystem 1 also absorbs light energy. You can see that here. And the energy or the electrons are energized and leave to be accepted by a different electron acceptor. So we talked about how that same process occurs in PS2, but this time it's just a different electron acceptor. Next, when these electrons are released, they will be passed down to the proteins that are listed here, such as FD and NADP plus reductase. And that will allow for the regeneration of NADPH from NAD plus with the addition of a hydrogen ion. And that will also be used to help fuel the Calvin cycle. All right, so this summarizes what we've talked about so far. We start here. We have solar energy that's going to come in to PS2. Remember, that's our first step here. And that involved having electrons be excited so much that they leave our chlorophyll molecule and attach itself to an electron acceptor. We don't go into detail about what those acceptors are. There's tons of them. Now, to replace that electron that was lost here, water splits to form 2 hydrogen ions and replaces an electron. And then we're left with the oxygen that will then be a byproduct and breathed out by the stoma or stomata. All right, so this plays a part in that hydrogen ion concentration gradient. Anytime you see brackets like this, that means it's talking about a gradient, or excuse me, a concentration. Okay, backed up here, our electron that that left our photosystem 2 and attached itself to an acceptor molecule will now be passed down to the electron transport chain. That electron transport chain will help provide some ATP after the electrons cause hydrogen ions to be collected on one side of the thylakoid space, and then that ATP will be used to fuel the Calvin cycle. Light energy is also going to penetrate the leaf and go into the thylakoid membrane to photosystem 1. Same thing is going to happen at the beginning as what happened in photosystem 2. We're going to have electrons, or an electron that will leave the photosystem to be bound to an electron acceptor molecule, and that will be passed down protein complexes that will help formulate ADPH from NADP plus with the addition of a hydrogen ion, which again the inner membrane space is full of because that was the whole purpose. There's a hydrogen ion concentration gradient. Okay, so that's what we've done so far. Now these byproducts, NADPH and ATP, are going to be used to fuel the Calvin cycle. Okay, and this just shows you one more picture of that. I'm a big fan of pictures, so whichever one makes you understand it better is the one you might want to study. But it shows the same thing. This is our thylakoid membrane, which is found within the chloroplast. And here we have our beginning phase here, where we have light energy coming in to photosystem 2. Which will have a result of energizing the electrons. One will be kicked out to a acceptor molecule that will be passed down our electron transport chain, resulting in lots of hydrogen ions moving into the inner membrane space. This demonstrates, not to forget, that the electron that was kicked out of PS2 has to be replaced by splitting water via photolysis. Now these hydrogen ions here will be used to fuel ATP synthase that will make ATP from ADP plus phosphate. And then also we have photosystem 1 that is harvesting solar energy, but the electrons that is kicked out from PS1 will be used to fuel the fire to produce NADPH from hydrogen and NAD+, both of which, NADPH and ATP, fuel the Calvin cycle. All right, and so that's what the first point here says. It's powered by ATP and NADPH that we generated in the light reactions. All right, so now we're talking about the Calvin cycle. This part of the process occurs in the stroma of the chloroplast. All right, so we're no longer in the thylakoid membranes. We're in the stroma, the liquid that fills the chloroplast. And the end product is glucose, C6H12O6. This process involves 3 steps: carbon dioxide fixation, reduction, and regeneration of the first substrate, which we'll call Ruby P. It has a long name, and I think I mentioned it in one of the upcoming slides, but we'll just call it Ruby P for short. Okay, so step 1, fixation of carbon dioxide. During this process, carbon dioxide comes from the atmosphere, the air, and it's going to come into the leaves via those stomata. Remember we said those are like tiny little plant mouths on the undersides of the leaves. And carbon dioxide, once it gets there, will attach to Ruby P. The fancy name for Ruby P is ribulose 1,5-biphosphate. So now you can probably see why I say we're going to just call it Ruby P. So it is able to attach to Ruby P because an enzyme is involved that we will call Rubisco. That actually stands for RUBP carboxylase, which would technically then be ribulose 1,5-biphosphate carboxylase. But again, for the sake of our sanity, we will call it Ruby P and Rubisco. So CO2 in the presence of Rubisco forms Ruby P. Okay, sorry, my handwriting's so terrible. I hold my pen much differently than most people do, and so it causes me to hit buttons on this pen that came with this device. So forgive me. All right, step 2 is reduction of carbon dioxide. In this phase, we're going to utilize NADPH that formed in the light reactions. We're going to use it for its electrons. We're also going to utilize some ATP, also gotten from the light reactions. And what's going to happen is we're going to form 6-glyceraldehyde 3-phosphate. We're going to call that G3P. G3P can be used to form glucose. However, in order to make this a proficient process, lots of G3P has to be made because not all of it's going to be converted to glucose. We need, for every, let's see, how am I going to explain this? During this process, 1 G3P can be made into glucose. But then 5 G3P that are made are going to be needed to reform Ruby P. We have to reform Ruby P because if we don't, the cycle cannot continue because it's a cyclical thing. It goes round and round and round. So we have to be able to regenerate our reactants in order to get the products over and over again. And it utilizes that NADPH as well as the ATP from the light reactions to do this. So the fate of G3P. Now we said most of the time it will be used to regenerate Ruby P. It will also be utilized to make glucose, but it can also be utilized to make other things. Sometimes G3P can be used to make amino acids, glycerol, fatty acids. It can be broken down into glucose phosphate, which can then be utilized to make sucrose. Sucrose is table sugar. Cellulose, which is found in plant leaves. It helps, well, as well as just plant cells in general, especially their cell wall. It helps them get a really rigid structure and be very prone to withstanding mass gains in height, for example, or droughts and even being full of water. So, and then it can also be utilized to produce starch as well. So the point here is that G3P can be utilized to form lots of different things. One example is glucose. Now, and the other, for every 1 glucose, I'm going to back up just so y'all can see it. For every 1 glucose, we're also going to need 5 of them to reform Ruby P. But again, it doesn't have to be glucose. It can be any of these other things here, just depending on the plant's needs and its capabilities. Okay, so this cycle here, or this picture here, demonstrates the Calvin cycle. And so again, it's a cycle, and that's why you see it in a circular format. So we have carbon dioxide that is going to come into the Calvin cycle, and we see the first step is CO2 fixation. Let's see if I can circle that. That's our first step. Second step was reduction, and then third step is regeneration of Ruby P. So again, the first step is carbon dioxide fixation. Second step, reduction of CO2. And then regeneration of UPP is last. And again, you'll have 5 of those that regenerate. You need 5 of those 5 G3Ps in order to regenerate Ruby P. And then the extra one, the 6th one, will be utilized to produce something, such as glucose or other organic molecules, starch, sucrose, cellulose, etc. Okay, so now there are some variations in photosynthesis in plants. Plants have been around for a very, very long time, much longer than animals, and they've adapted several different methods of being able to withstand their conditions in their environment, which is really important for them because plants aren't exactly modal. You know, they can't get up and walk away if they're in the sun and it's getting too hot for them, or they can't, you know, go drive to the next city over where it might be raining and they need the water. You know, they're kind of stuck where they are. So in order for them to thrive, they've come up with different adaptations to allow them to thrive in those types of climates. So when the weather's hot and dry, many organisms or many plants utilize what we call C3 photosynthesis. Most plants are C3 plants. And they're called C3 plants because a 3-carbon compound is formed first after carbon dioxide fixation. During this process, the stomata close. That prevents water loss. Okay, so when the stomata are open, the plants are subject to water loss because water can evaporate from the plant, and of course that's not good for the plant. They need water. However, closing the stomata also impedes the exchange of CO2 and oxygen. So remember, the primary purpose of these stomata is to breathe in, quote unquote, carbon dioxide, but also to release oxygen in addition to preventing water loss. So this impeding of that gas exchange is going to reduce the efficiency of photosynthesis because carbon dioxide and oxygen are going to be building up within the plant leaf. And they'll have to compete for Rubisco. And so less glucose will be made. But again, it's kind of like, you know, the pros for the plant outweigh the cons. If the plant dries out due to leaving the stomata open, it'll kill the plant. So they close their stomata, photosynthesis efficiency is reduced, but at least the plant can live. Okay, another variation in photosynthesis are C4 plants. So again, preventing water loss is critical. And in C4 plants, they do things just a little bit differently. So closing the stomata to limit water loss, like we said, also limits CO2 intake and allows oxygen to build up, which can be bad for the plant. So some types of plants do C4 photosynthesis. This is where a C4 compound will be formed first after the fixation of carbon dioxide. That just means C4 just means it's a 4-carbon compound. So another thing that's the Calvin cycle and the light reactions are not occurring simultaneously in the same space. So we have partitioning of the pathways. So the chloroplasts and the bundle sheath cells carry out the Calvin cycle, whereas the mesophyll cells can fix CO2. Okay, so this slide here shows you the difference between a C3 and C4 plant's anatomy within the leaf structure. So this is showing you the inside of the leaf. Here you can see the stoma or stomata. This is called mesophyll tissue. You can think of it kind of like the meat of the leaf. And then here we have the veins that would run through the leaves. C3 plants have mesophyll cells that arrange themselves parallelly, like that. And the Calvin cycle and the light reactions, everything occurs within these cells. In C4 plants, however, the mesophyll tissue or cells are going to arrange themselves concentrically around the leaf veins. So we can see that here in a circle. And then within that circle, we have these bundle sheath cells. And that's important because we said that, you know, carbon dioxide and oxygen, when the stomata are closed, can build up within plants. Well, oxygen is toxic to cells when it's in high concentrations. So these sheath bundle cells protect the cell from buildup of oxygen while still allowing for the mesophyll cells to fix carbon dioxide only. So in this way, the C4 plants are able to partition the pathways in space, meaning it's compartmentalizing it, where some parts of the photosynthetic process will occur within the mesophyll cells and some will occur within these bundle sheath cells in order to prevent toxicity to the cell. It allows them to be more efficient at photosynthesis. All right, so again, C3 plants carry out both reactions, the light reaction and the Calvin cycle. within the mesophyll cells. That's an advantage in moderate conditions. But these C4 plants, they have adaptations that allow them to be better suited for environments that are really hot and have really dry weather. That allows for the stomata to stay closed. That allows for conservation of water while avoiding oxygen overexposure, which can be toxic to the cell. Now, the last variation we'll discuss is CAM photosynthesis. So CAM stands for Crassullian acid metabolism. A lot of plants that you might think of in desert environments, like cactuses, succulents, they are involved in CAM photosynthesis. They partition in time rather than in space. Let's go back for a second. One more. Here we said that we are partitioning pathways in space. Only certain reactions will occur in mesophyll cells versus the bundle sheath cell. That's partitioning them in space. In CAM plants, we are partitioning in time. So during the nighttime, CAM plants open their stomata when it's nice and cool outside, at least compared to the daytime, and they use C3 molecules to fix carbon dioxide. That forms C4 molecules. And then they store these C4 molecules in vacuoles and wait till the daytime. And during the day, they keep the stomata closed to avoid water loss. They release stored carbon dioxide when NADPH and ATP are available from the light reaction. Additionally, we have some more evolutionary trends that we see in plants. These C4 plants, they're most likely evolved in and are adapted to areas of high light, high temperature, and limited rainfall. They're pretty sensitive to the cold. C3 plants do better than C4 plants below the temperature of 25 degrees Celsius. And CAM plants, they compete well with C3 and C4 plants when the environment is extremely arid. Think, like Arizona, for example. So CAM plants are pretty quite widespread. All right, everybody, that was chapter 6. I hope you enjoyed it.